Let me hit record here. But what if I want genetic variation? Mitosis doesn't get us that, okay? Mitosis doesn't give us that at all. That's where meiosis comes in, okay? Now, I am not going to ask you the steps of meiosis in terms of describing it. We're going to go through it all, but you'll start to see, as we start to explain it and look at it, we're going to see a lot of similarities between this and mitosis. Now, if I was a gambling man, I would pay attention to the stuff that is different, okay? When it comes to the differences between meiosis and mitosis, I may ask you to explain some of the differences between, because we're going to see there's the same steps occurring: prophase, metaphase, anaphase, telophase. But now we're going to see there's an additional cycle of this, okay? Whereas mitosis, it goes through all those, cytokinesis, bam, one cell becomes two identical. A little bit different here with meiosis in that regard. I don't know why it is not letting me. There we go. The great thing about this, and I have a few different numbers there, and I'm not going to ask you the numbers, okay? Because some of the numbers you're not even going to know how in the world to say, to be honest, because I had to look them up because they're so huge and so fantastical in number that it's really hard to really put your mind around it. But there are, 70 trillion different genetic combinations between the mating of two individuals. And when you start to look at some of the potential things that can even inject more diversity within that genetic combination of mom and dad, we even get these even huger numbers. So the great thing about this, it is this first bullet right there. It introduces a huge amount of diversity, okay? We're going to talk about it in very general aspects today, but probably, and I doubt we'll get to it today, so we're going to talk about it in general today. But when we look at how males and females form their sex cells, their gametes, and these are just sex cells: egg, sperm. Okay. They vary greatly. We'll hint at some of we'll look at some of the processes that, spermatogenesis and oogenesis. We'll talk about those. We'll describe them a little bit. We won't get into a whole heck of a lot of detail though. But when we look at just when we start, for us guys, it's pretty straightforward. It begins when testosterone starts to skyrocket. Puberty, 12, okay, maybe 11, but 12, 13, whenever that is for a guy. And usually that's around 12. And females, though, females are complex, okay? And I'm just talking about formation of their eggs. For females, they start producing a whole batch of them in utero when they're developing inside mom in the womb. Then they pause. And then nothing happens again with those until they reach puberty, which can be anywhere between 10 and 13 when they start. Then they'll go through another phase of meiosis and stop. And they only finish it up if pregnancy occurs, if fertilization occurs, okay, if the sperm gets to the egg. So when it comes to females, there is a lot of variation that occurs there. And because of that degree of difficulty, if you will, they start with a huge number of them, and then they whittle them away, and to the point where they have, a few million different eggs in their ovaries, in utero, and then by the time they get to puberty, they only got a few hundred. So females are much different in that regard, but we'll hint at some of that when we get to it. So let's look into all this overview of meiosis, if you will, okay? This is a special type of reproduction, of cell division. This is sexual reproduction, okay? This only involves the sex cells. Only involves the sex cells. Only sex cells. like Anthrax says, only, okay? Only one. We're going to see similar terminologies. I mean, you know, there is going to be some bit of mitosis occurring here. It just does, okay? We're going to start with some stem cells, and these stem cells are going to have to duplicate and increase in number, and that's a mitotic type of a thing. Yes, it should. Celeste, yes, it should. So let's see. What if I do this? Keep, cancel. Okay. I screw up anywhere else? Yeah, god dang. Okay. Chromosomes still replicate. Chromosomes are still going to duplicate inside these cells, okay, as these sex cells start to mature. So as the egg goes through its phases of development, as the sperm goes through its phases of development there, when we go from the stem cell to the cell that's actually going to enter the phases of meiosis, it's going to duplicate. Okay, so it's going to do all that stuff that it does in S phase. Okay, so nothing's different there at the very, very, very start. Still the same. The purpose of meiosis is to give a vast amount of diversity, yes, but the next major goal of meiosis is to half the material. I have to half the material because if I don't, I have way too much genetic information. And if it's way too much genetic information, the cell can just abort. All right. The cell will stop itself, it'll rest itself, and it won't continue the fertilization process. Okay. So we got to get this from what's called a diploid. Okay. And 2N. And 2N is just, it has the full complement of genetic material, all 23 pairs or all 46 individual chromosomes. Meiosis, again, produces haploid sex cells. And this is sex. Whoops. Whoops. Freudian slip. Sex cells. Okay. So I have to get this genetic material to be half, because half from mom, half from dad, get you. Okay. So if there is a defect in how the cells divide, one cell could have way too much. And if we have an excess chromosome because a chromosome didn't separate properly, that can get us monosomies and trisomies and nosomies, aneuploidies, okay? It's called aneuploidies, where you don't have any genetic material, okay? The chromosome's missing. A classic one is trisomy 21, right? Down syndrome. So there are potential defects that can happen, and we'll talk about that probably on Monday, because I don't think we're going to get through all of this today. Okay. These gametes, these sex cells, the egg and the sperm, fuse to form a zygote. And this is going to primarily happen within that fallopian tube in the female, if you didn't know. As soon as mom and dad's N come together, boom, we have our 2N structure, and this thing is going to become you eventually. So we're going to be talking about chromosomes, but in meiosis, we are going to be talking about homologous chromosomes. Okay, homologous chromosomes. Homologous chromosomes are similar. Homo means same, right? These have the same length. Their centromeres are in the same place. It's just that one is coming from mom and one's coming from dad. And we call that paternal and maternal. Okay. And we know they're homologous. We know they're the same because when we take a picture of it, a karyotype, you can see they have the same banding pattern. Okay, same banding patterns, same banding patterns. Yep. Okay, yep, all same. They're all the same. Okay. Yeah, chromosomes occur in pairs. 23 pairs of chromosomes or 46 different individual ones. I usually say 23 pairs because that's just what rolls off the tongue. Okay. But the great thing about this, they're going to have the same length, the same centromere. One comes from mom, one comes from dad, but they are coding for the same thing. It's just that on mom's chromosome and on dad's chromosome. And what does that produce when mom and dad's chromosomes come together? Well, it depends on the trait. And we'll start looking at that nonsense in chapter 12. Okay, in chapter 12. With how genetic traits can be passed from one generation to the next. Because not everything's a mix. Okay, not everything's a mix. Sometimes one dominates, sometimes one goes to the background, sometimes it's a blending, sometimes it's codominant, sometimes it's multiple alleles at various different chromosomes. So there's a lot of variation there when it comes to those traits. But the homologous chromosomes, moms and dads, the same chromosome, same size, same shape, coding for the same information. Just one's dad's version of it and one is mom's version of it. And that's what we're going to be talking about here. Okay. They have genes. Genes code for a protein. Code for a protein. Okay. Proteins make things. They make structure, right? They make things. Okay. Could be a structural component. Could be a some kind of protein receptor for the cell membrane. They make stuff, right? That's their primary function, is to make things. When we make a protein, okay, it's going to be exhibited in whatever that string of amino acids tells that protein to become. Okay. And like I said, dad's got a version, mom's got a version. Okay. And as you can imagine, we have a lot of genes within the population, and there is a bunch of variations for it. Okay. Look at skin color, look at hair color. Okay. A number of variations exist for hair color, eye color, right? That gives us a lot of variation. So mom has a copy, dad has a copy, and that gets you, right? They can encode for the same thing. Same dark colored hair on mom, same dark colored hair on dad, same light colored hair on mom, same light colored hair on dad. Or they could be different. Mom's a blonde, dad's a brunette. Okay. But it's still the same gene at the same location. They're homologous. Okay. So it's still the gene for hair color. It's just that mom's is light and dad's is dark. Okay. We look for genes, especially when it comes to, and we're going to be talking about this in chapters 12 and 13, I think it is. No, 12 and 14. Okay. We're going to be looking at genes and how the cell reads the DNA. Okay. How the cell finds the gene for it. There's protein markers within it, within the DNA. So it'll find the protein marker that codes for the gene for hair color. Okay. It's the gene. Okay. Well, we call this thing an allele. Okay. And we'll talk about how we represent alleles more in the next chapter when we start looking at Gregor Mendel and these genetic factors, okay, that went into passing inheritance. Okay. So we don't use the word gene for whatever reason. We call it alleles. And alleles are what we represent on a chromosome. And in chapter 13, in chapter 12, I believe, 12 or 14, I can't remember. They're both pretty similar. I have a link to a website that you can go and you can find every single thing in our chromosome and what that gene codes for. Okay. And we use alleles because it's easier to put big T, little t at a spot than write out receptor for hypercholesterolemia, H57, hypercholesterolemia, and genetic variant 1, right? So we use what are called alleles. Alleles code for a specific gene. Alleles are for a specific gene, okay? Gene for hair color, we'll use big B, little b, okay? If they are both the same, okay? Let's say black hair is dominant to blonde hair. Pretty obvious, right? What if we say black hair is dominant to brown hair and brown hair is dominant to blonde hair? So that gives us an allele population of big B, big B, big B, little b, little b, little b, blonde, brown, and black hairs. Okay. If they are both the same, they're homologous. If they are different, we are going to refer to them as heterozygous. Hetero, different. Homo, same. Okay. They use short fingers, long fingers. Okay, sure, I could have used that too. Okay. We get one from mom, we get one from dad. On those chromosomes, they have information for various different genes. We call these things alleles. Alleles are what come from mom and dad coding for a specific gene because, I mean, this doesn't tell me jack squat, okay? If I have this and this, I don't know what would be made, okay? Don't know what would be made. I need both of these, that and that, to get me an outcome. If I don't have both of those, then there's going to be a different outcome. It's not going to be what should normally be made. And with different genetic conditions, which I believe is at the very end of this, we get into some of this, okay? We get a different outcome, okay? You get a hallmark appearance, okay, due to these genetic defects, okay? And some of them are very innocuous, you wouldn't even know, okay? Some of them have a very hallmark appearance to them. And that's how they're kind of initially diagnosed until further studies are done. So here is a nice little picture that shows the overview of meiosis. Okay. Again, this is a reduction division. Okay. This is a reduction type of division. I'm going from full complement to half. Half from mom, half from dad. Okay. As you can kind of see here, I get 4 haploid daughter cells. Okay. This is not mitosis. Okay. I don't get an exact copy. I don't have the parent cell giving rise to 2 cells. All right. I have an individual cell that starts it off, the parent, the P1 generation there. And it goes through 2 sets of divisions. Okay. It goes through a phase of meiosis 1 and a phases of meiosis 2. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. It's the basic same things occurring. Okay. We're just going to add a slight little twist on it. We're going to introduce, I think, 3 or 4 words. Okay. But the point of meiosis is to get this sucker down to the haploid. Okay. Cutting the info in half. And give me 4 haploid daughter cells in the process. So if we look at kind of this overview of meiosis 1, as we'll get into all the individual steps. But if we look at this overview, okay, chromosomes are replicated prior to meiosis 1, just like mitosis, right? We had that phase of interphase before the phases of mitosis. Same jazz here, okay? Nothing different. Each chromosome doubles up because I'm going to double it up and then I'm going to cut it in half, right? Homologous pairs of chromosomes line up. We call this thing a synapsis. Okay. As you can kind of see underneath it there, they may recombine, they can exchange. We'll get to that here in a second. Your homologous pairs line up at a metaphase plate, and then they separate. Okay. So there's a prophase happening, there's a metaphase happening, there's an anaphase happening, and then they pull apart from one another, and I have 1 cell becoming 2, and there's your telophase. In meiosis 2, I don't have an interface. Okay, there's no real. You can call it cytokinesis. Not too many books do. Some call it cytokinesis. Some just call it the separation of 1 cell into 2. Some cells do it. Some cells don't. There is some degree of variation here depending upon who you are. You're an animal or you're a plant, yada, yada, yada. But again, they are now in the phases of meiosis 2. And this is going to look very similar to mitosis. So if you know mitosis, you're basically describing meiosis 2. Except I'm bridging in a couple new words. That's it. Same dang steps. Same dang things occurring. It is the same thing. Oh my gosh, sister chromatids, right? Sister chromatids, right? Sisters, okay? And then these things are going to separate, right? Just like they do in mitosis, but now it's meiosis. Dos, okay? And we get our 4 daughter cells with 1 chromosome in each. Haploid.